Have you executed your task? You know I'm not joking. Eric?
Sorry, coffee comes first.
I know.
Yes.
What did you accomplish?
I had a couple of really good conversations.
Turn this around.
Yes.
Did you have good conversation?
I had good conversations, but most importantly, I was able to direct someone to an entrepreneur to have a conversation with them.
Very good. And now we're going to be throwing this out in this direction. Let's see, in the back here, please. Catch.
You're throwing.
No coffee in the
People
room.
with coffee.
No coffee in the room. No coffee in the room. That's why. Who picked it up? Who picked it up? Any good conversations?
Yeah, I had very interesting conversations, and I discovered that possibly we have partners here, and that was great.
Good to know. Good to know. Thank you. So I said this is a sporting event. I don't know if that would qualify. I'm throwing this at you. Good conversations?
Yes, I continued ongoing conversations with some parties that we've been working with for 2 years.
I like it. I like it. All right, now that you're warm again, this is important because this panel is not about, certainly not about me, and it's obviously about the panelists, but we want it to be an interactive panel. I will kick it off with some very basic questions, but the complicated, tricky questions have to come from you. So the panel should be known to everybody, but just in case you just walked in the door, or what was the expression, Ulrich, have been hiding under a rock or something? I don't know who does that. Who hides under a rock? Laura from Panther, Ulrich from Torque, Professor Hammond of the Hammond Lab at the Koch Institute and MIT widely, and then this gentleman is John, and he works at the ILP and represents corporate relations. We are here to debate what is happening to the field of cancer, its current state, its immediate future, and its long-term prospect. So that's a very tiny thing we're going to accomplish today here. I'm going to start off with a very simple question. Where are we with cancer innovation today, Laura?
We have still a long road to go. Like, cancer has been on the headline for a very long time, and many improvements have been made, but we have still not been able to cracking down. So there is an enormous amount of work to do, both from the engineer, the physician, the biologist, to understand the disease. Like, there is plenty of room for everybody to throw their hat on the table.
Well, that's good. Ulrich, what is your view?
Well, I think cancer has changed dramatically in the last few years. There's always been this notion that the immune system was holding cancers in check. I don't think most people believed it, that to cure cancer you had to kill the tumor cells, but not engage the immune system necessarily. I think we're starting to understand the interplay between the two, even how chemotherapies will engage the immune system, and that's part of the mechanism. And then obviously immuno-oncology is making a tremendous impact as well. But there's so much more we don't understand now than a few years ago.
So the actual object of study is also changing with that. That's what you're saying. It's expanding
it's
and
much more of a
contracting.
multicellular, and physiology kind of challenge now.
Professor?
Yes, I think along with immunotherapy, which is providing these really exciting opportunities, there's been an extension of the work going on in understanding different cancers on the molecular and cellular level. And that leads to a much greater ability to develop personalized medicine. I think we're seeing people beginning to bring machine learning tools, as well as data analysis, big data, and, I don't know, medium data, different levels of data sets that can actually inform therapies. This means that if we can understand this both in the tumorigenic space and the immunogenic space, we can actually develop therapies that are directed toward smaller groups of individuals. This is something that's always been a topic. Well, it's been a topic for the last several years, but I think it's becoming more of a reality because we're beginning to access meaningful data. What has to happen, though, is that we have to have more access to larger sets of data, which means much more agile means of getting samples from patients.
So let's, can I, if I may just pivot back on that question a little bit. It's interesting, like in traditional medicine, the problem has been that the N is usually 1 or 8 or 10. So there's no big data, there's no medium data, right?
Right.
But with genomics, presumably, you now, each person, and tell us a little bit about how this changes, because how can you really do machine learning on medicine? I guess that's
You can
really
only
the
do
question.
machine learning on medicine if you have data sets that allow a meaningful approach. You need something to learn on.
And why have these sets emerged so quickly over the last just few years?
I think that the sets themselves are just beginning to emerge. I don't think so quickly is probably what everyone would agree to. But the fact is that there have been some larger scale projects, especially since the human genome studies, that have enabled us to build libraries of genetics, for example, for ovarian cancer, for example, for breast cancer. That's the beginning, but it's only a peak, and it's a small cross section. What we need is coordinated data. And what we have not had is access to data across hospitals and across countries, and to get that level of data sharing, that capability to understand what the genomics are of different cancers, we need a much deeper level of collaboration. Then we can begin to really unleash the machine learning tools. But people are beginning to develop those tool sets.
John, let's bring in the,
Sure.
the industrial side, but you have been a bench scientist,
so
you've been in pharma.
I'm in this awkward position of representing industry, which I haven't been in for a while, but I'm at MIT now. But before I get to that, I think I would just want to add, it's when you reflect back on 7-10 years ago, that data you had no access to it, period, because everything was in Scribble. The data wasn't even being collected. And now we're talking about how to actually collect that kind of data in a way that it can be made available and to be processed. But in terms of where, I think the question was where cancer research
Where is it now?
Where is it
basically
now?
this entire debate is.
So I guess I would say
from
from the industrial standpoint, again, if you go back 15 years, virtually every pharmaceutical company, maybe they had made the move into biologics, but mostly the activities were around coming up with small molecules that you could administer systemically, essentially poison the cells more than you're poisoning the patients, but of course the patients are getting tremendously poisoned. And there's been an explosion over probably the last 10 years, but it's kind of accelerating. I think we're in the middle of an explosion, not anywhere near the end of it. And frankly, it's exciting and uncomfortable at the same time. Like, you can't know it all, and so you have to have things like this where people with different backgrounds can communicate with one another.
So John, then, when you think about the corporate partnerships we have here at MIT in this field, what, if anything, has been happening, and is happening now? Is there an increased vigor and an increased, I don't know,
Sure.
even number of connections that are being made right now as opposed to 10 years ago, or is
So
this
I
sort
can't
of-
really reflect back on that 10 years ago since I wasn't here, like at least not from the perspective of MIT. But I was saying earlier in a conversation that we've gone through this, industry has gone through this period of thinking that you have to do everything internally and that you can collect all of the external information that you need through patent searches and literature searches and maybe some connections that you have through conferences or relationships that your employees have with professors that they've worked with in the past, all the way to we're going to put most of our discovery outside, or we're going to put most of our even aspects of our development outside. And in the initial stages of that, it was just buy up all of these assets, buy up all of these access to services, and that transitioned into a place that we are now pretty rapidly because that resource dried up. All of the companies were looking for some of the same things, to where we have real relationships, real funding of fundamental research, and in terms of the relationship with startup companies, it isn't waiting for that proof of concept and then it goes to the higher bidder. It's getting involved sometime earlier. Industrial investment in actual projects that can change the direction of the startup.
I was just picking up on what John said. I've seen a huge amount of increased interest from industry in engaging with academic institutions and nonprofit institutions early on to collaborate. And I think this collaborative spirit is something that has definitely grown. And it's happening earlier, not downstream, but upstream of the development of a new therapeutic. If you combine that with some of the new tools that we have, we have CRISPR and ways of understanding genetic profiles of cancer. We have ways of addressing more directly genetic changes. We have different ways of delivering siRNA and mRNA. All of these things are coming together to make more possible, and that requires this level of collaboration upstream. We begin to talk about how we can use those tools effectively.
So let's talk about that in detail at some generic level that we can all kind of communicate across, I guess, scientific and non-scientific boundaries. So I'm just going to summarize what the 8 startups told me, so you'll see how little I know. So Travera, right? So they're engaged in finding a predictive biomarker, which is interesting. So that's another that's a buzzword. So I think the aim of this next section is I'll name these buzzwords that I picked up through these 8 startups, and I'll try to see, and I'll be asking you, what, if anything, is missing from this picture that the audience should keep in mind. So think about that. So biomarkers, right? So there's a functional test for cancer, is what Travera is doing. Honeycomb is doing single-cell profiling, and they're claiming this is an advance in precision medicine, especially targeting protein and RNA. You, at your lab, Professor, the Hammond Lab, is doing multi-layering of various medicines. So that can have also a transformative effect on the entire system. We'll talk more in detail about what you're doing in the future, because you were alluding to some things there that could perhaps also be commercialized. Panther, that's you, right?
That's me.
You were talking about local drug release and the importance of the local tumor site. And that was also mentioned by another startup as a very interesting concept. Electrify, again, here is talking about IV infusions, accelerating the delivery of cancer drugs themselves, including the timescales. So there's something here, as Professor Hammond said, about the delivery mechanism, the speed, the cost probably as well of the delivery. And then Sextant is talking about harnessing cell death to create immunity in those cells that are going in as part of the medicine, presumably or as shoring up basically the cells before they go any further. And Leucolabs is talking about the ever-present risk of infection, right? So the white cell monitoring, even at a patient-by-patient basis in an outpatient situation. So that again changes the whole trajectory of now we're not just in the lab anymore, we're actually delivering something to the primary physician and maybe even directly to the patient. So here we're talking consumer medicine. And then lastly, Asimov, right? Potentially a massive breakthrough in genetic circuit design, enabling an operating system for the body, and an operating system that's synthetic biology, which I already thought was pretty fast and advanced, but could use to accelerate further changes, perhaps at the level of the Genome Project. Maybe I'm wrong about that. So these are some of the things that were just mentioned today over a 20-minute period. What's missing? What else is in the mix?
Oh. Did not hear that one coming.
This is the homework. This is the homework.
Yes.
So tell us. So this was 8 startups.
Yeah. So I would say that probably the use of big data for target analysis, for target identification. Radiology is probably missing. There are some huge advances in radiology. Probably people from Philips know about that well, where you can actually target the tumor and the shape of the tumor using MRI imaging, and even if the person is moving, you can change the beam. And
bring some other ideas in. What else is in the picture here? Colleagues of yours, friends of yours starting companies, colleagues at the Koch. What else is actually happening at the forefront of this work? What other big sort of systemic change technologies and techniques are now being developed over the last 2-3 years? Is there anything else that we should be aware of, that we have missed?
that you have missed, but you described pretty large. I'm trying to think. So I have colleagues who have also been involved in very careful cellular level measurements. We talked about single cell. That's one area. There's also being able to measure even the mass of a cell and how it changes and how that might be used for detection. We didn't talk a lot about imaging, and there are some fascinating things going on with imaging. Now, for example, if you take advantage of second window near IR and you use appropriate probes, we may be able to detect tumors that we weren't able to detect before, including metastatic tumors. That would change the way we address diagnostics, but also treatment. So I think that there are some enabling tools there that we haven't talked about. There's a lot of work going on, for example, in the Belcher Lab, and being able to advance those technologies. I'm trying to think of what else you haven't named. It's hard to do the subtractive thinking, by the way.
It is very hard,
yes.
Which is why we have a fantastic panel. Ulrich, so you are up to T cells. What else is in the mix around your colleagues, the people you're talking to?
I think going on the topic of imaging and understanding things in multiple dimensions, you know, when I was trained in sort of classical pharmacology, we talked about compartments and, you know, multi-compartment models to a 2 or 3, right? You had the blood, the tumor, maybe the brain, right? That was so crude, right? To really understand these systems, we have to go to a more microscopic scale of different compartments and how they interact with each other physiologically. And then there's a whole framework, and that was a big part of what we did at Merrimack, was bringing computation into biology. We don't talk about that so much as a discipline, but it needs to overlay much more what we're doing in terms of understanding not single biomarkers relationships, but multiple biomarkers, you know, in what goes on in multiple dimensions, et cetera.
I have a specific question on that, Ulrich. Are we now at the stage with the science that someone who is, let's say, in a completely different field, say a top computer scientist, can come in and deliver very, very useful breakthroughs even without knowing the bench science?
I don't think so. I think what we heard going on in synthetic biology is going to get us there, where people with that kind of skill sets can plug right in. Traditionally, what we saw, especially at Merrimack, but also when I was at MIT, was getting people to work, you know, mathematicians work with biologists is really hard unless they have an understanding of the biology or the math. That interface doesn't really work, but, you know, things like synthetic biology is going to change that.
I want to maybe just do...
Incidentally, everybody here is nodding. I'm thinking, of course they can work together. But
well, but it's an interesting question. Maybe you have an observation on this, and I think Anne was alluding to this as well, the whole Koch Institute creation, right? So 10 years ago, and it took, I mean, I don't think it took 10 years. There were results probably year one, but, you know, what we're talking about is not easy. Like, you can talk about collaboration across disciplinaries, you know, but you guys have done it. I mean, you know, you're in a startup that probably is doing this.
Yeah, so we, on the collaboration part, especially with the computational model, we are trying to integrate everything at once. Like, we have the understanding of the disease, we have the understanding of how the drug works, and one of our basic strengths is, can we deliver it better? So we brought in mathematical people that can predict the distribution of the drug. We know the drug very well, we know the tumor very well, we know how we want to deliver it. Can we predict how far into the tumor goes, how far into the other organs goes? And that's not a conversation easy, like, because there is the mathematician and the biologist, they speak two different languages. So you need either to train them at some interface or bring someone in the mix that is versatile enough to serve as an umbrella connector.
So I'm going to build on this, but after my next question, I will be throwing that ball around, the catch box around, just so you know. So what I'm interested in from the panel side is for you to reflect on how you arrived at what your current focus is, and try to kind of make a reflection that also illuminates what path people should take if they're thinking about bringing a thought to commercialization, so the part of your work that has brought, you know, either the company out or brought you kind of to the commercialization forefront. What was it that brought you there? Why did you commercialize this particular technology, or why are you thinking about that? So maybe we'll start with you, Ulrich Schlörer just spoke, and then.
Oh, that's a tricky question. I mean, I've always been interested in the interface between engineering and biology. That's why I came to MIT in the first place as a postdoc. I thought that would be the right place to do it. That was 16 years ago. I arrived here, and none of that was happening, I'll be honest with you. But it slowly started to happen, and I was part of a big DARPA grant with like 30 people, and I think 5 or 10 of us started working really well together, and many of those at the Koch today as professors and other things. So I'm always looking for those opportunities where engineering meets biology. Merrimack was that, where it was a combination of computational biology and understanding cancer. TORC felt like a very similar problem. And material science meets immunology. Yeah, that's where I see I can bring something to it, right? That's also what you're looking for as an entrepreneur. Yeah.
Who wants to go next? Would you want to go next?
No.
Paula?
All right. So the first part of that, the collaboration, is something that was really critical. And I'm a polymer scientist who works on material science problems. And my own lab actually evolved quite a bit because a large portion of our lab had been focused on electrochemical engineering and batteries and fuel cells. So when I became a part of the Koch Institute, we had just begun to look at some early studies of how to generate material systems that would be interesting for delivery. Being a part of a community that is planned and designed around collaboration does make a huge difference. And in my lab, the big movers in that were not just myself, but my lab members. They were immediately, especially on moving within the same space, incredibly interactive with each other. And you know, we mentioned the language earlier, and I have to agree. We spoke different languages, and those students and postdocs rapidly learned new languages and learned how to speak to each other. I think if you were asking what needs to happen for these kinds of translational things to take place, you need to create environments where people are having constant collisions with each other. So it's not enough to simply have a department of X and a department of Y, but bringing people together in collaborative teams and forcing them to articulate their ideas in ways that all of their colleagues can understand.
That must be painful.
It can be extremely painful in the beginning, but extremely rewarding. So now we can find biologists who can talk to an engineer without using 20 acronyms, and an engineer who can talk to a biologist without going into all kinds of device talk. It becomes incredibly enabling.
I think he also fosters creativity. Like it's like if you don't speak a language and you are thrown in a country where they speak a different language, you need to be creative. So as an Italian, you usually start working with the hands and trying to make people understand you.
Does that help you?
It helps.
It helps.
It helps. You will, following my mom advice, you can also just scream and shout, and they will understand Italian better. But I don't think that works. But it kind of forces you to make sure other people understand you. Yeah. So you come up also to a way of explaining, like, looks like a pie or looks like give example, makes you look things in a different perspective and say, oh, actually that's true, it looks like a pie, so I can do these other things.
Okay.
So it fosters creativity.
Sounds good. So did you have something you wanted to say on that? Why you ended up where you are?
Based on the question, I'm not the right person to answer, but I would comment on one thing that the innovation, a number of people have said it in different ways, innovation is really a pedestrian sport.
Right.
And the closer up to a point, the closer that you are together, the more you'll have those sorts of collisions. And this is really an ideal environment in Kendall Square because there's such a density.
Yes.
Not only of biological sciences, which is extremely important for advancing biological sciences, but those other areas that are tangential and are increasingly becoming part of what biological science.
So let's invite some questions, and please, any question is allowed. It can be about sports, it can be about anything. Try to keep it on cancer. All right, now watch this.
Good throw.
Okay, I think a couple of weeks ago, I was at DC for a immunotherapy, I think NIH have a special session for the immunotherapy from the mathematicians and the physicists. So they ask, what kind of computational, mathematical, approaches can be employed to help the immunotherapy, approach. So, I mean, bunch of physicists or mathematicians, they present some work. But my impression is that it's kind of very, very, preliminary, stuff. Just curious, like MIT at a Koch Institute, is there any intense plan, to do some heavy, computational approaches using the mathematics, or
There must be an answer to this question.
Yes, the answer is yes.
Is there an investment in computation at MIT?
The short answer is yes. It's just getting started, but there are early partners already involved in efforts that involve industry. The newest one is a new collaboration with IBM, but you're going to see more of that over time. There is an intentional effort to bring artificial intelligence to the biology and chemistry and molecular interfaces, and it's taking place across institutes.
There was an answer to it. And let's start here.
Hi, so I was just curious a little bit more about how you feel about older technology. So you see a lot of companies now working on CRISPR and immuno-oncology, but there's older technologies that may have been slower to develop products or get into the clinic, and they may not be as sexy to investors anymore because they might be all these older ideas. Can you think of any technology that you think people have kind of forgotten but there's still hope for? And how would a company working in one of those technologies actually be able to make themselves seem attractive to investors or partners?
All right. I don't know who that question is for. Anyone can pick it up.
Old technologies.
So I think that new technology are great. They also take a long time to be developed, be accepted, to go through the FDA process. So there is some benefit in old technology to be reinvented. But you need the innovation. You cannot just, even old technology, they need to thrive in something new. And I think we are an example of that. We don't have any new CRISPR drugs. We don't have micro robots that goes anywhere. We are innovative in the way we approach the problem and in the solution that we give. But we are using surgical procedures that are currently the standard of care. So when that was one of the problems while we were at MIT, we were not, this was one of the projects that was not on the top of the innovation. Like, we don't have self-assembling material that, we are using grass material that are already into the clinic. So we are old, but we are reinventing the old to make new waves of success into the clinic. And that investors likes, because the timeline for us to enter into the clinic, it's way shorter than a CRISPR molecules or a new path. So we are their first step while they still invest into the CRISPR. Maybe they will put more money into there because there is more development that needs to be done. But if old works, you need to innovate, and I think there is hope.
Paula, what is your observation? Ulrich, you have an observation?
Yeah, I'll just say that the older technologies, right, become the foundation for what we're building on. At TORC, without what's happening in protein engineering, liposomal science, and cell therapy, we couldn't put all the bits and pieces together. And I think it's the same for CRISPR. CRISPR is great, but you still got to deliver it to cells to have an impact, or you got to apply it to cell therapies or whatever.
And I would say echo the same thing. I would prefer the word known technologies to old technologies. And I think known technologies can actually be very helpful. So in the company that I co-founded, Layer Bio, we take something that is known, which, for example, may be an implant of some kind, and we coat it with our system, which is new. So just by the nature of the work that we do, we're building new on top of known to generate a system that can more readily get into the marketplace than if we were starting with something that was entirely new. And it does help with getting translation across, but it also helps to be informed by a technology, to have a path because of that, and to use that as a way to ladder in new technologies and the work that so hopefully we'll be starting a company. My desire would be to start a company based on the layer-by-layer nanoparticle platform and to find partners and somehow leverage that in some way in the future. And again, we'd have the known plus the new combined, because we would use the generally regarded as safe liposomal platform and build on it.
All right. Let's do quick here and then on. Question. Question. Question's got to be a question. Only questions.
Well done.
Thanks. That's going to hit me in the head. I've heard a lot about intervention. What is new in terms of prevention? And has there been anything in terms of profiling environmental factors versus genetic factors? And has anybody tried to look at the difference? Or is there a difference? I mean, cancer is cancer, right? But
oh, you got one of the things that was missing from our list.
Yes,
Yeah.
you did the subtractive thinking.
She did it. All right, so prevention. Tell us about that.
I think the big one we were missing was the microbiome, which is much more directed at prevention than I think treatment of disease. If you really look at the fundamentals, how bacteria in your gut can influence what happens in the immune system and all kinds of places in the body.
So it took the audience to answer the question, because it was a trick question, because I knew the answer, because we had a synthetic biology event, much like this, with none of the same startups, just 2 months ago.
I'd say another example is
So it was just a trick question. You always got to put
I
a
would say
wedge in
another
there.
example is anticipating pathways to resistance and being able to get a read on that very early, so that the treatment that is followed does not actually lead to specific resistance. It moves over to something else before that happens.
Sort of following on your question, or actually I had my question already in mind, but it's a nice follow-up. Early detection as a form of prevention, right? Because if you detect it early enough, then you can actually prevent the deadly part of the disease. So if you think about ovarian cancer, one of the deadliest forms of cancer, if it's detected at stage 3 or 4, it's a 30% chance of survival. At stage 1, that's up to 90%. Guess how many people are diagnosed at stage 1? A whole whopping 15% of people. So if we can shift that, we could have a huge impact on disease. And so one of the things that really bothers me in my professional life is how little money there seems to be for diagnostics for early detection. And does that mean it has to come from academia? Do you have any sort of insights into how we might change that, or how we might make it something that people pursue in pharma, startups, as well as academia?
I guess it's more a question for industry in that case.
Oh, that's me.
That's you.
Supposedly. Yeah, so I guess you're referring to things like companion diagnostics. I mean, in terms of business models, companion diagnostics that would help performance of a drug. I think there's a reluctance to invest in those sort of things because it's very difficult to make a profit, typically off of diagnostics. Even the high throughput kind of assaying that some you would call them diagnostic companies are doing, it's really hard for them to make a lot of money. There seemed to be a lot of promise maybe 10 years ago when genetic testing was something that was very expensive, and so you could have some very expensive assays and invest in technologies for being able to run them. But then the cost of actually doing genetic testing just went down and down and down, and so there isn't so much profit to be made there as well. So I don't have a solution to it, but I mean that's part of the challenge that I hear, especially when pharma companies are talking about going into diagnostics. That how do we actually make a product out of it that benefits the patient and the company?
We have a bunch of questions. I'm going to be challenging 2 smart guys from Philips next, but I'm going here now.
In terms of diagnostics, I find your response interesting. Grail, which spun out of Illumina this past year, raised a Series A worth just north of $1 billion. So you're suggesting with a $1 billion Series A, there may not be sufficient profit to generate a return on investment. Liquid.
Well, I guess I'm talking about different things. You're
yeah, I think we are
starting a
talking about
company,
different
and I'm
things.
talking about products that you
right
would in-license so that you can.
Right. But this is a liquid biopsy approach with GRAIL, and it was basically this issue of early detection sits right in the square of that.
Right.
And early detection essentially amplifies the benefit of every therapy that's out there. So I think there has to be a way to find a route to profitability.
So countering everything I said and really answering Anna's question is to have many examples of that.
Yeah. I was just going to contribute that insurance payors are the parties that could actually profit from prevention.
Absolutely.
And if you incentivize them to save money so that they can detect a cancer and not have to treat the patient, they make money. So they're a key player.
All right, so we're edging towards the making money part. We're going to get to Marcia. You got a free pass for a second?
So I just kind of related to this. I was curious what you guys have seen in developing new diagnostics. Like, what are some of the new technologies that will improve diagnostics?
New technologies, improving diagnostics.
There are so many. Liquid biopsies were mentioned, so detecting circulating tumor cells in a range of different ways is something that has advanced incredibly in the last 8 to 10 years, probably less. There are ways in which one can design probes of different kinds that can essentially elicit a response from a tumorigenic cell that causes degradation and a signal, for example, in urine. There are epigenetic ways of determining that tumorigenic cells may be present, and diagnostics are being developed around being able to bind specific epigenetic signatures of cancer. I'm going to play to my neighbors.
There are temporary implants that do that have some kind of detection and readout. That's an area that's had some interest in the recent years. But I would echo that. I mean, liquid biopsy seems to be something that the attraction to that is so huge. You avoid some kind of procedure. You perhaps can even do it in your own house, but if not, you can do it in a clinic that doesn't have a huge amount of infrastructure.
So, Phillips, I'm going to allow Brian even a comment, because I was so mean and teed up the question, But what are you guys up to that is relevant to this?
I mean, we're up to a lot of things, but let me talk about what was kind of top of my head. One fantastic event once again. I mean, it's really nice to be part of this community. It's a unique place in the world. It's in Trond, and the ILP office does just a fantastic job. Wish more people were here. We got to promote these things more, because again, I think it's bringing people together like this, just bumping into each other, that really makes the difference. But what's top of my head is, I mean, the discussion today was fantastic, and if you think 5-10 years out, we're going to be creating these amazing solutions to cancer. But how do we do a better job as a community bringing that to every cancer patient on the planet, right? We don't want to solve cancer just in Boston or in the United States. What could we be doing as a community to pave the way for rapid adoption so that everyone, whether you're in Africa, whether you're in Australia, China, Europe, how can we get this to everybody?
Okay, easy question. How can we get it? How can we get it to everybody?
So many of these technologies won't go to everybody immediately, at least. There has to be a long development in manufacturing and even regulatory, to some extent, process to bring those prices way, way down. I mean, there will be instances where people come up with something that is, fast, more effective, and cheap, but it won't be all the time. So it's a
Can you
long
bypass
process.
that? We have 2 startups here. Can you bypass this? I'm hoping you're going for rapid and quick and cheap. You know, isn't that what startups all go for?
Yes, that's what we aim to go for.
That's what you say to investors?
Well, they will tell us there is the FDA in front of you, so you can go faster than you can.
Yes.
So there is but I think something that's missing on this point is the power of the patient's population. We haven't been really touching about that, but we have seen that we are targeting pancreatic cancer, for example, and the community has been so supporting for us. Like there is all of these patients advocacy group, there is the connection that can help you to reach different geographical area and spread the technology and the collaboration with different hospitals. There is a need from patients right now in cancer, I think, to really take ownership of their disease and looking at what the opportunities are. We have been reached out by email by the doctors, but the family, by the patients. They want to be informed for what the technology does, even if we are not yet into the clinic. So I think that the strength and the power of patients is not to be undermined. There has been some instances where patients' advocacy have been able to force the hands of FDA. That you can argue it's good or bad, but there have been cases where drugs have been given for compassionate use, and that has been resulting in proof into the clinic that helped the technology to move forward.
So as we're rounding this up, you were burning with some comment.
I'm sitting here trying not to be a buzzkill. And so there was another opportunity to be a buzzkill. So I want to actually say something positive. I'm trying real hard. So another way that, and it happens at MIT as well, but a lot of companies have done this, is engage directly with potential customers in the countries that you've named and try to come up with technologies that can serve at the price point that those countries can handle, or the people that they're talking to can handle, that can function in the environments. And that's a very direct way of solving some of the same problems, or maybe less of the problem, maybe 80% of the problem. But something that's very beneficial to companies that are doing this is oftentimes that spawns an idea that they can bring back to, let's call it, the developing world. Not that Australia is not part of the developing world, apologies if I made that implication. Lots of backpedaling. Lots of backpedaling. I've totally lost my train of thought. But
I think that where we are moving and where I want to move the panel is to the future. So that question is about the future. Where is the cancer ecosystem moving in the next few years? Because we've been talking about these 8 startups. We've heard from the Hammond Lab, and we've heard from industry interaction. But where are we moving next as an ecosystem? So this is Cracking Cancer 2.0. We had a summary last December, and I don't really remember enough to pinpoint what's so different. I need to go back and think about that. But what would you see, Cracking Cancer 3.0, even just 1 year from now, if I asked you about what's emerging? Is there a sense that this system is moving into and shaping and morphing into something different, or are we more or less seeing now the building of a platform, and that's going to take a while? Easy question again. Let's go to Ulrich for this one.
I mean, I'm a little biased here. I think cancer therapy in particular will go more towards curing patients, which changes a lot of the conversation. Also about how to bring this to the rest of the world. It's very hard to see how you treat metastatic cancer in Africa who had 2 months survival and needing a ton of supportive care. But if it's a one-shot kind of bedside manufacturing of a cell therapy, that might start to make sense, at least in some places. So I think we'll see more of that. I think we'll see more of prevention too, as a big part of the dialogue, also for society as an economic thing.
Paula?
I actually think that I agree with the movement to try to cure cancers, try to expand the number of cancers that can be addressed using the immune response, right? And this is something that's a real challenge, but if we can get inroads into even a few more of the more advanced cancers in that area, then we're going to have a huge impact on the number of people who die from cancer. I do think that we talked about data and computation and machine learning. I think that will begin to find its place in the cancer world, and we'll be talking about it more. And I'm a chemical engineer, so I'm not naturally the person to talk about this, but because we are focused on precision medicine, and I think there could probably be something of understanding the tumor immunomodulatory environment, that also has some precision aspects to it, that these will begin to converge. And we will begin to have ways of honing in on targets more rapidly.
One
So-
other part,
Go ahead,
sorry.
go ahead.
One other part on the where we are going is that cancer has been always seen like cancer is only one disease, and right now everybody is going to the understanding that's not the case. There is different cancer type, but also within the same cancer type, there is the microenvironment actually does a lot to it, and so there is a curve that's starting right now to actually understand the biology at the single level for the single cancer type, and that will spin out in different technology 3 years from now, but there is.
We're going to end this panel with, we can have a short question and then I will prime the panel, please. So before this question, I will be asking you for the single thing that you're the most excited about right now in the field of cracking cancer. And you get one word.
I was going to mention
one word.
vaccines. There are already vaccines for cervical cancer. There are companies developing shared neoantigens in cancer. Those could potentially be vaccines which themselves are very cost effective.
All right, so summarizing, one word, what are you the most excited about right now? We can start with John.
Precision. I'm implying another word.
It's good. Paula?
Oh man, you stole my word. Okay, precision vaccines. I can add a word.
Got it. Ulrich.
People start daring to use the word cure, which didn't happen years ago.
Ooh.
And the multidisciplinary collaboration.
All right, so between precision cure and multidisciplinary collaboration and a few other things we've talked about, including CRISPR, data, methods, collaboration, drug delivery, radiology, moving to impact, computation as such, early detection, vaccines, and moving beyond just talking about systems like blood, brain, and tumor, and moving into more specific things, and including the power of the patient community. I think we have summarized today's discussion. Please go out and make partnerships happen, and see you soon. Bye-bye.